Welcome to the Deep Dive. Today, we're diving into ecological restoration, specifically thinking about the UN's big push, the decade on ecosystem restoration running through 2030. It's a massive goal. And a huge part of restoration, especially in really fragile places, is tackling invasive species first. We're talking about islands. Invasive species are probably the biggest single reason for biodiversity loss, even extinctions on islands worldwide. But managing them, it's incredibly tough. These places are remote. The logistics are, well, often a nightmare. And just knowing if you've succeeded, that's a whole other challenge, financially and practically. So today we're digging into something that might be a bit of a game changer. It's a paper called Environmental DNA for Invasive Rat Surveillance and Biosecurity on Islands, led by Sebastian Steibl and James C. Russell. They're essentially asking if we can use water samples to figure out what's happening with rats on land.
Exactly.
Yeah.
It tackles this core problem island managers face constantly. Can we take this tool, environmental DNA or eDNA, which has been amazing for tracking things in water? Can we actually adapt it to reliably keep tabs on invasive species on land, specifically rats, in these really remote island settings? They're looking for something faster, cheaper, and crucially reliable for that ongoing island biosecurity job.
Okay, let's unpack that a bit because, eDNA usually makes you think fish, maybe whales, stuff living in the water. How does DNA from a land animal like a rat end up there?
Well, it's actually pretty straightforward when you think about how water moves.
Yeah.
Even just minimal contact droppings, urine, maybe hair, skin cells gets washed into the environment. These animals are shedding DNA constantly, and islands, especially smaller ones, are kind of perfectly set up to collect it. Small islands, particularly these low-lying atolls like in the study, they have these huge coastlines relative to their area, a really large circumference to area ratio. So basically, everything funnels outwards. Rain, any small streams, even groundwater moving towards the coast. It all carries this terrestrial DNA right into the lagoon, or maybe into inland lakes if they have them. The water becomes this passive collector, almost like a giant sponge soaking up biological signals from the land.
Right, the water's giving you the clues. And the paper looks at using the signal for two really critical moments in island management. First, just figuring out what's there before you even start trying to get rid of them, which rats are present.
And second, the really tough part, checking if any survived after an eradication attempt, or if new ones have shown up, that whole biosecurity monitoring phase.
So to test this properly, they needed a good location. They went to Tetiaroa Atoll, it's in French Polynesia, and they set up a really rigorous study design, what they call a before-after control impact design, or BACI.
Yeah, that BACI design is important. It lets you compare things properly. They specifically chose islands with different known situations. One island was already confirmed rat-free, had been since 2020. Another island just had the Polynesian rat, Rattus excellens. And a third island had both the Polynesian rat and the ship rat, Rattus rattus, living together. So 3 distinct scenarios.
Okay, and how did they actually collect the data? What was the method?
It was pretty systematic. On each of those islands, they collected 40 water samples each time they visited. 20 samples from the beach shoreline, facing the lagoon, and another 20 from the edges of any brackish or freshwater lakes on the islands. They did this once before any rat eradication work started, in May 2022. Then they went back twice afterwards, 3 months later, and then again 9 months later.
40 samples per island each time. That sounds like quite a bit of sampling. Was there a specific technical hurdle they had to overcome for this?
Absolutely. The really crucial bit, the innovation here, was developing a brand new test, a novel Rattus-specific metabarcoding assay.
Okay, metabarcoding. That's basically scanning a sample for DNA from lots of different species at once.
Exactly. You use specific genetic markers. But here, they needed something really precise because the 2 target species, Rattus and Rattus excellens, are actually very closely related genetically. So the assay, the test, it had to be sensitive enough to reliably tell them apart, even if their DNA was mixed in the same water sample or if DNA drifted from one island to another nearby. That differentiation was key.
Right, makes sense. So you've got the setup, the new tool. Let's get to the results. Before the eradication, when rats were definitely there and likely abundant, did the eDNA actually pick them up?
Oh yeah. For confirming presence when rats were common, it worked really well. Impressively well, actually. Depending on the island and the specific rat species, somewhere between 5%. And get this, up to 90% of the individual water samples came back positive for rat DNA.
90%? Wow.
And importantly, that new assay they developed, it did its job perfectly. It could successfully tell the difference between R. rattus and R. excellens, even when they were on neighboring islands.
Okay, so it's reliable for saying, yes, rats are here, and here's which rats. But what does that actually mean for someone managing these projects? How efficient is it compared to, say, trapping?
That's where the modeling they did comes in. It shows some pretty dramatic potential efficiency gains. Based on the detection rates they found, you'd only need somewhere between, say, 2 and 65 water samples to be 95% sure that rats are present before you start an operation.
Hold on, only 2 samples in some cases.
Yeah, for Rattus eDNA detected on the beach samples, the model suggested just 2 samples could give you that 95% confidence level. It was a bit higher for the lake samples, maybe 25 samples for R. rattus in the lake environment. But still,
That's incredibly different from traditional methods, right? Setting out dozens of traps or cameras for days on end.
exactly. Think about the fieldwork time. Collecting those 40 samples per island, the paper says it took about 4 to 5 hours. That's it. Compare that to setting traps or chew cards, which need at least a couple of days on site, usually more.
And the cost. Islands work is notoriously expensive.
Right. They estimated the total cost for an eDNA survey on a typical 100-hectare island would be somewhere between roughly $2,000 and $400 to $250. Now compare that to camera trapping, which they estimate at $3,000 to $6,000, plus you have the time cost of analyzing all those images. Or detection dogs, which might be $2,250 to $4,500, plus daily operating costs and transport. So eDNA looks very cost competitive, potentially cheaper. The big saving is likely reducing the time experts and gear need to be physically present on a remote island, which is often the most expensive part.
You mentioned lab work too, PCR replicates. What's the efficiency story there?
Yeah, PCR replicates are basically rerunning the genetic test in the lab on the same sample extract, just to be extra sure you're detecting real DNA and not some contamination or error. It adds time and cost.
Yeah.
What they found was really encouraging. Usually only 2 or 3 replicates were needed to confidently detect the rat DNA if it was actually in the sample.
So not endless retesting.
No, which again saves lab time and money. It really strengthens the case for eDNA being efficient for these initial baseline surveys confirming presence before you act.
Okay, it sounds fantastic for finding rats when they're common. Almost too easy. But we know the real nightmare for island managers isn't finding lots of rats, it's finding that one elusive survivor after an eradication. Did the eDNA hold up in that biosecurity phase after the main operation?
Well, this is where the story gets more complicated.
Yeah.
So after the eradication attempt, the local rangers did eventually confirm through intensive island-wide monitoring that, yeah, small numbers of Polynesian rat survivors, R. excellens, were still present on 2 of the islands, hiding out probably.
Okay, so rats were definitely still there, just in low numbers. What did the eDNA show?
0. Nothing. Both the sampling done 3 months after and the sampling done 9 months after. They got absolutely no positive detections for rats from the water samples.
Wow. 0.
Yep. And what's more, they also ran traditional monitoring baited trail cameras for 72 hours along the exact same shoreline transects where they were collecting the water samples during that 9-month follow-up.
And the cameras?
They also failed to detect the survivors. 0 detections there, too, along those specific transects.
Okay, so 0 detection from eDNA, 0 from cameras right there. But we know rats were on the island. That sounds like a pretty major failure for using eDNA for biosecurity, for confirming absence.
It certainly highlights a huge challenge. But the authors argue it's maybe less a failure of the eDNA tool itself and more about the difficulty of sampling design when dealing with extremely low densities.
We mean?
Well, when rats are everywhere, pre-eradication, their DNA is likely widespread, easy to pick up. After an eradication, you might only have a handful of survivors left, and they're not evenly spread out anymore. They're probably in a few hidden pockets, maybe moving around. It becomes very patchy.
So the water samples and cameras were just in the wrong place.
Essentially, yes. The study notes that the survivors were eventually found using much wider, more general monitoring methods, often more than 300 meters away from the specific beach and lake transects used for the focused eDNA and camera comparison. It underscores a key point. No matter how sensitive your detection tool is, whether it's a water bottle for eDNA or a high-tech camera, if your sampling plan doesn't put the sensor where the target actually is or where its signal is concentrated, you won't find it, especially when targets are rare and patchily distributed.
That makes sense. Sampling design is always critical. But wasn't there another issue? Something about false positives on the clean island?
Yes, that was another really interesting and slightly worrying finding. On the control island, the one confirmed rat-free since 2020, they actually got 5 positive detections for R. excellens DNA from the beach samples.
5 positives on a supposedly rat-free island. How?
The suspicion, and it seems quite plausible, is physical transport of eDNA fragments. They think that wind and waves carried tiny bits of DNA across the lagoon from the nearby islands where our exoens were present.
Carried across the water. How far are we talking?
Potentially over 2,000 meters. That's more than a mile. These DNA fragments might have just washed up and accumulated on the shoreline of the control island.
Wow, okay, that has major implications for using eDNA for biosecurity, especially if you have islands close together.
It really does. It suggests that eDNA isn't just staying put. It can move, potentially quite far, in water or maybe even carried by sea spray in the air. So if you're managing Island A, which is rat-free, but Island B right next door still has rats, you might constantly detect rat DNA washing onto Island A. It makes confirming true absence extremely difficult in those situations. You have to be really cautious interpreting a positive signal.
So a powerful tool, but with some serious caveats, especially for that post-eradication check.
Definitely. But let's also touch on the versatility advantage they highlight.
Right, because unlike, say, a detection dog trained only for rats.
Exactly. With eDNA, the fieldwork collecting the water stays the same regardless of what you're looking for. The difference is all in the lab work, downstream. You just adjust the genetic sequences you're targeting. So you could use the same water samples to screen for rats today, then screen them for invasive ants tomorrow, or maybe monitor the recovery of native seabirds the day after just by changing the lab assay.
That adaptability is a huge plus for broader conservation goals, not just single species eradication.
It really is. It opens the door to more holistic biodiversity monitoring using the same sampling effort.
Okay, so let's try and summarize the big picture here. What have we learned from this deep dive? It seems eDNA from water samples is definitely a strong contender, maybe even better than traditional methods for that initial step. Confirming if invasive rats are present and which species, especially when they're relatively abundant, it's rapid, it's cost-effective.
Agreed. Big ticks for presence absence in high density and for species identification. The fieldwork efficiency is a major win. But the reliability takes a nosedive when you get to low densities, the biosecurity stage. That's partly down to the challenge of sampling design for rare targets.
And partly this issue of eDNA fragments getting transported between islands, causing potential false positives on supposedly clear islands.
Precisely. So for managers, great tool for initial assessment and prioritizing which islands need work, but probably not yet reliable enough on its own to declare an island officially rat-free after an eradication, especially if there are nearby source populations.
So what needs to happen next for eDNA to really live up to its potential in this area? What's the future research direction?
Two main things stand out from the paper. First, we desperately need a better handle on how eDNA actually moves around in these coastal environments. We need to understand and model this transport, these eDNA plumes. How far do they spread through water, maybe even through the air? Knowing that risk zone around an infested island is critical for interpreting results correctly.
Okay, understanding the movement. What's the second thing?
Technology. Right now, you collect your samples, and then they have to go back to a specialized lab, often shipped internationally from these remote places. That adds a significant time lag.
Which kind of negates the rapid advantage of the field work.
Exactly. The real breakthrough will come with the development and application of robust, portable sequencing technologies. Imagine being able to analyze the water samples right there in the field or on a support vessel nearby, getting results in hours instead of weeks or months. That would truly revolutionize the rapid response capability for biosecurity.
Field-based analysis. That would be a game changer. So this work clearly shows eDNA is a powerful new lens, especially for confirming presence. But it leaves us with a really fundamental question, doesn't it? When you've potentially reduced an invasive population down to just a few scattered, wary individuals, how do you even design a sampling strategy, eDNA or otherwise, that has a good chance of finding them? That challenge, reliably detecting extreme rarity, seems like it's still the absolute core problem for confirming success in island restoration. And maybe eDNA, despite its promise, hasn't quite solved that one yet?